Hello! In this series of videos, I'd like to break down the process of the central dogma. This is an amazing process that gets the information held in the base sequence of DNA and uses that through a two-step process of transcription and translation to make a protein that folds into a specific shape and fulfills a function within the cell. So in this first video, I'd like to talk about the regulation of transcription. So transcription, as you can see here, is the process of making an RNA copy of one strand of a particular region of DNA called a gene. So in this video, we're going to focus on how this process is regulated. The DNA in every cell of your body is exactly the same, but obviously there are different proteins in each cell that help that cell fulfill a specific function. So for example, even though you have the genes in your skin cells that determine your eye color, those genes aren't turned on in your skin cells. The genes that are turned on, meaning transcribed and translated, are ones that are important for skin cell function. So let's look at the DNA sequences and proteins that are important for the regulation of transcription, meaning transcribing only certain genes when, where, and how much they are needed in cells. So whenever you see a box in these videos, that's going to be a DNA sequence that's important. I've zoomed in on just a small portion of a DNA sequence here so we can use that. And whenever you see circles, these will be proteins that are important in this process. So the first thing that we need in order to transcribe a gene is the enzyme, the protein that actually does the transcribing. And that enzyme is called RNA polymerase. In eukaryotic cells, RNA polymerase is brought to a gene at a sequence called the promoter. So the promoter is a DNA sequence that's specifically recognized by proteins called transcription factors that bring RNA polymerase to the start of a gene. This +1 here is going to indicate the first nucleotide that's going to be transcribed. So notice that RNA polymerase doesn't actually bind to the gene at the +1 nucleotide. It binds what we call upstream of that at the promoter. And it's brought there by these proteins called transcription factors. Now in eukaryotic cells, this is actually not enough to transcribe a gene. We need additional DNA sequences that are sometimes very far from the genes that they regulate and proteins that bind to these genes. These DNA sequences that are important for regulating when and where and how much genes are turned on are called switches. These switches are bound by proteins called activators that can, like their name implies, activate RNA polymerase to start transcribing a gene. So to see how this works, you can see in this slide here that what happens is when activators in pink bind to switches, that facilitates bending of the DNA molecule such that these activators can interact with transcription factors and initiate RNA polymerase, which is shown in green in the slide below, to start transcribing a gene. So you need all of this to transcribe a gene in a cell type. And the reason that only certain genes are transcribed in certain tissues is that these activators are only present in those tissues or cell types. So for example, if this is a gene that is necessary for skin cell function, you would find in skin cells the activators to turn on this gene. And you would not find in liver cells these same activators. And the activators become active and bind to the gene in response to signals from the cell's environment. So you need all of this to begin transcription of a gene. And what happens when transcription begins is that RNA polymerase is going to make a single-stranded RNA molecule that's going to be complementary to one of these strands of DNA. And it's going to be complementary specifically to the template strand. The other strand of DNA is actually not going to be used at all in this process. So it's called the non-template or coding strand. So if we go back to our drawing and look here, I can actually, based on the polarity of the DNA molecule and where I put this promoter, I can tell which of these strands is going to be the template and which is going to be the coding strand. And the reason I can tell this is that the template is always read 3' to 5'. So the template strand is always going to be read 3' to 5', and so the non-template strand is anti-parallel to that and is going to run 5' to 3'. Now if RNA polymerase sits down the promoter here, it's going to transcribe this gene this way, and it's going to transcribe the template strand. So we can tell here, based on the polarity that I've indicated, that the bottom strand is going to be used as a template because it runs 3' to 5' left to right here. The top strand is going to be the non-template, or sometimes called coding strand. So what happens when an activator binds to a switch and transcription factors bring RNA polymerase to the promoter is that the two strands of DNA are separated by RNA polymerase, and RNA polymerase is going to make a single-stranded copy of RNA that is complementary to the template strand. So when RNA polymerase sits down at the promoter, it's going to start transcribing at the +1 nucleotide. So here is going to be this transcribed region of the gene. Now a gene actually encodes both, a gene actually includes both the transcribed region and the promoter, just by definition. So RNA polymerase sit down on the gene and start transcribing at the +1 nucleotide. And the RNA that's transcribed is going to be complementary to the template strand. It's also going to be anti-parallel. So the template strand being read 3 to 5 means we're going to make RNA molecules in the 5 to 3' direction. So let's go ahead and do that. I'm making an RNA that is complementary to the bottom strand here, the template strand. And remember that there are U's in RNA that are complementary to the A's in DNA. There are not T's in RNA. Keep going, keep going, keep going here. So this is what RNA polymerase is going to make. This is called our transcript. So we know how RNA polymerase finds a gene and starts transcription. The promoter is important for that. How does it know where the gene ends? And the sequence that's going to be important for this is called the poly-A sequence. This is going to become important later in the RNA transcript, but it's also important for signaling RNA polymerase that it's reached the end of the gene. So RNA polymerase is going to continue transcribing the template strand all the way until it reaches the poly-A sequence. And this is kind of a signal that says, hey, you're done transcribing, and sometime directly downstream of this, RNA polymerase will fall off the gene, and we have what's called a primary transcript here. And in our next video, we are going to talk about the signals in this primary transcript and how this primary transcript gets processed before it gets translated. Thank you.